Hello friends, welcome back to my YouTube channel, The Zone of Med. Yes, the name of the channel is changed from Zone of Anatomy. It is now Zone of Med. So as you see already in the thumbnail, today we are going to discuss about complete general physiology that contains our cell organelles, homeostasis, and the junction, or we say the cell communication, intercellular communication. So let's proceed it. First of all, let's see what is a cell. We say cell is the structural and functional unit of life. So when we come to its function is very much specific. Then moving forward, its total cell in our body is 100 trillion and the RBCs is 25 trillion. Coming to the similarities, like our body has many different type of cells, but there are some similarities which are similar in all those different types. What are they? First, in all cells, oxygen usually react with carbohydrates, fats, and protein for what? To release energy required for all functioning. Second is chemical mechanism. So what is this chemical mechanism? The general chemical mechanism is basically for changing nutrients into the energy, are basically we say that is similar in all cell. Coming to another point, that the all cell delivers their end product of their chemical reaction into the surrounding fluid. Then coming, almost all cells have their ability to reproduce, that is to form new cells of their own kind. And another one is, suppose if a cell of a particular type is destroyed by any means, for example, if there is any trauma, so the cells get destroyed, the remaining cells of this type usually then regenerate. So the cells show power of regeneration also. Then we come that in our body there is extracellular fluid and intracellular fluid, but the most important one which we need to remember, that extracellular fluid is 1/3 of our total body and the remaining one, that is 2/3, is of our intracellular fluid. So our total body fluid is 60% of the body weight. Moving forward, what is extracellular fluid? So we say the fluid outside the cell of the body is called extracellular fluid, as the name suggested. So when we talk about the our extracellular fluid, so we say extracellular fluid is also known as the internal environment of the body, or we say milieu interior. So it is basically, the scientist who give this concept is known as Claude Bernard, and according to his theory, he say that all the all the internal environment in an organism is placed in a greenhouse. So the cell he considered it as a greenhouse. Make it as an MCQ. Then the components of internal environment given by him was cell, tissue, ions, and nutrients. So you need to remember extracellular fluid composition and the components lie within it. And when we talk about intercellular fluid, in intercellular fluid, we only need to remember one thing, that is a definition, and another is its composition, that is 2/3 of the total body fluid. That's it. Then moving forward, in our body system, we come across a term, that is our homeostasis. What is homeostasis? Basically, a homeostasis is the maintenance of our constant environment. What does this constant environment mean? It means external and internal environment of the body remains constant. This concept was given by one of the great scientists, that is W.B. Cannon. Learn the scientist name, it might come. So when we talk about how the body maintain its constant or homeostasis, so we say that organelles as well as tissue of the body maintain homeostasis. For example, we can say our GIT tract provides nutrition to our body. Then the buffer system, body buffer system, kidney and respiratory system together form their acid-base balances. We see now further lectures how it is maintained. Then another one is kidney maintain constant ionic concentration. When we talk about our kidney, we go through how it functions, and then we come about that the lungs provide oxygen to the extracellular fluid so that it can replenish the oxygen used by the cell. So these are basically the important parameters of homeostasis, or we say the important functions of the homeostasis. When we talk about the homeostasis, one word is basically come into our mind, that is feedback. Now let's talk about what is this feedback is. Basically, the feedback is the situation when our output for an event in the past will influence the occurrence of the same event in the present or a future. So let's see what happened in this. Suppose we have an auto response of a system to an impulse, which is received by that system. We call it as feedback, and the feedback can be negative feedback and the positive feedback. When we talk about the negative and the positive feedback, let's describe what is this negative term means. First of all, make it clear, in our body is very negative. Like we say be positive, but our body is negative. Like the most of the control system of our body works on the principle of negative feedback mechanism. So our body prefer negative feedback mechanism. In your MCQ, it might come which feedback mechanism is more effective, or most of the control system is controlled by which feedback mechanism. So answer is negative and not the positive. So the negative feedback is the tendency to reduce the input signal that caused it. Now let's see what is this negative feedback is. Suppose if some factors become deficient, okay, the control system plays a feedback mechanism, that is negative feedback mechanisms, which usually consist of the series of changes that return the factor towards a certain value. For example, in this, the example we take is our blood pressure. So how this blood pressure is maintained? Suppose there is a high blood pressure that causes the reactions that promoted a low pressure. If we see like carbohydrate metabolism, insulin and glucagon, we can easily distinguish this mechanism. Now, if we see another one, that is our plasma concentration. So if the plasma CO2 in extracellular fluid is increased, then automatically our respiratory centers increase. If our respiratory center stimulated, then our pulmonary ventilation increases, and so therefore the output result will be decreased in the partial pressure of CO2 in extracellular fluid. So basically negative feedback is done for if something is going high or some parameter is raising, then this feedback mechanism come into play and it lowers that. That's why in this fashion our body maintains our constant environment. Now let's see another one. Here we say that there in negative feedback mechanism, another one is water balance and electrolyte balance. So in exam you just need to know the definition. Second thing, their examples. Another thing you can see over here, that there is no viscous cycle. What this viscous cycle is, basically does not cause the death of an organism, whereas when we talk about the positive feedback mechanism, it causes the death. Now let's proceed towards the positive feedback mechanism. So positive feedback mechanism is very easy. That is the tendency to increase the event that caused it. So how it causes, we take an example. Suppose if a person suddenly bleeds out approx 4 liter or we say 3 liters of blood, okay, then automatically his cardiac pump started working low, as a result of which his blood pressure also decreases. As a result of that, if you think, the blood flow of the heart muscles will decreases, for which our heart automatically becomes weak because it is pumping very rapidly. Then further cardiac pump after some time becomes weak and further it try to lower our blood pressure, and in this fashion the complete cycle goes on, repeat and repeat, and ultimately it causes the death of an organism. So therefore we say that recovery of the heart pump is caused by negative feedback. But if we talk about the positive feedback mechanism, it is a viscous cycle, because if the heart becomes weak and weak after continuation of this cycle, then the person ultimately leads to death. And we say that the death is caused by the positive feedback mechanism, whereas if our heart pumping recovers, that mechanism is due to negative feedback mechanism. So another point or MCQ, that is the recovery of heart pumping is caused by negative feedback mechanism, whereas the death is caused by positive feedback mechanism. So write this MCQ. Coming to the positive feedback mechanism, the important positive feedback mechanism is childbirth. How it is childbirth? Suppose when in the uterus are contracted, so the baby's head pushes towards the cervix, and eventually it stretches the cervix and it sends the signals to the uterine. When we talk about these signals in the reproductive system, we come to know what type of signals was there, and once these signals goes to the uterine muscles back to the body, then it causes more powerful contraction. As the contraction goes on increasing and increasing, the process ultimately leads to a parturition of the baby. So in the childbirth, during the parturition, it positive feedback mechanism is very essential. Then when we talk about the generation of nerve impulse, it is also important one. And the final one is the blood clotting. How blood clotting? Because suppose if suppose we get our blood vessels get ruptured due to some accidents, okay, so the clotting factors are activated. We already know that clotting factors come, platelets, forms and everything, fibrinogens, clotting. So when we talk about this, basically you learn these complete process, that is platelet formation, in your immune system chapter. So till now you just need to know, if a blood vessel is ruptured, then blood clotting factors are activated, and some factors among that act as like some inactivated factors act on active factors and they activate it. So it is a complete process of activation of the clotting factor. So it's a separate topic. So no need to go in a deep. Just now you need to remember, in the blood clotting, the positive feedback mechanism takes place in childbirth, positive, and in generation of nerve impulse, it is positive feedback mechanism. So just remember these examples. It is very important. Nothing more to memorize among these. Another thing you need to remember is the components of the negative feedback mechanism. First of all, we need to know a set point value. What is the set point value? It is a center of the normal range, and which is treated by the control system as a target value. So set point value is also known as the target value. Then we have the sensors. What is sensor? To the the continuously monitor the control variables. The sensor is basically, continuously monitoring the variables that are fluctuating. Then we have the comparator. The comparator basically initiates a counter response. And effectors, the mechanism that store the set points in the normal value. So set point value, that is our initiator, and effector that usually set point to its normal value, whereas the point or set point value is the target value. So do not confuse between target value and the normal level. Now we have discussed every basic details about this, and the examiner will ask only this, like they can ask that the viscous cycle is. So the viscous cycle is present in a positive feedback mechanism and not in negative feedback mechanism. Then we talk about the role in homeostasis. So like we say, positive feedback mechanism is sometimes be useful, but the negative feedback mechanism almost control most of the control system of the body. So do not forget these points. Now we are saying control system. So we have a 10 control system of our body, and we have already learned about this. Just quickly recap: cardiovascular system concerning with our heart, blood vessels and blood, and its function is to transportation of material throughout the body. Digestive system consists of our GIT, liver, pancreas, and its basic function is assimilation of nutrients and elimination of waste products. Then endocrine, we have the endocrine glands, that is coordination of body function with the help of hormones. Then immune system, thymus, spleen, lymphatic system, blood vessels, defense mechanisms. Integumentary system, that is our skin, which protects us from the external environment from harmful rays. Musculoskeletal, skeletal muscles on the bone for support and movements. Nerves, brain, spinal cord, and peripheral nerves. Coordination of body function with help of the electrical signals. Reproduction by gonads, penis, vagina, uterus for procreation or the formation of new born babies. Then respiratory system, lungs for the exchange of gases, carbon dioxide and oxygen. Urinary system, kidney and bladder for the homeostasis of ionic concentration. Now, we are done with this all. Now moving to the cell. So we say cell is a structural and functional unit of the living organism. Now, when we talk about the cell organelles, we basically divide our cell into plasma membrane or the cell wall and the protoplasm. Protoplasm gets subdivided into cytoplasm and nucleus. And then when we say that the cytoplasm consists of the cytoplasmic organelles and non-cytoplasmic inclusions. So cytoplasmic organelles can be non-membranal organelle and the membranal organelles. You need to know what are membranal organelles and what are non-membrane bound organelles. Direct MCQs are going to come in examination. So learn this by heart, especially this yellow-blue box. And then this complete flow chart. It is very easy for you, but the most important one is this part. Cell membrane. So cell membrane, we say it is a thin, it is elastic structure which envelops the cells. And we termed it as cell membrane, plasma membrane. When we talk about the structure, we follow fluid mosaic model, which was given by Singer and Nicholson in 1972. It is basically a lipid bilayer, which is containing with the large globular protein molecules. We all know these basics. Coming to the membrane lipids consist of our phospholipids, glycolipids, and cholesterols. Then we already know that it has a it has two parts, that is head part and the tail part. The head part is polar, that is water soluble and hydrophilic, whereas tail part is non-polar, that is water insoluble and hydrophobic part. So these are the basic things which we have already done. Just go through because the MCQ comes in this polar hydrophilic head part. So I mentioned over here. When we come to the membrane protein, we have one line with us, that is they float like an iceberg in the sea of lipid. So the sea is considered as the lipid bilayer and the iceberg is equivalent to our membrane proteins. Coming to the membrane proteins are integral proteins as well as peripheral proteins. Integral proteins are completely passed through it, like it do not completely embedded into it. It is not partially. So integral are completely passed through membrane, and its function is adhesion molecule. It helps in pumps, It helps in carrier. Then it acts as receptors, enzymes, or we can say when we try to elaborate it. So adhesion molecule that anchors cell to the neighboring or to the basal laminas. When we talk about pumps, we have an active transporting ions across the membrane, sodium, potassium pumps, and many channels we have. Then carriers, we have facilitated diffusion. At that time, we talk about these carriers. Then we have ion channels through which ions can pass through into it. Then we have receptors that binds our hormones. When we talk about in the membrane physiology about G protein coupled receptors, so we come to know what are the hormones bind to it, what neurotransmitters bind to it. So it is important. Enzymes, then enzymes are basically catalyzing reaction at the surface of the membrane. Then we have our antibody processing and distinguishing self and non-self, basically concerned with our immune system. Peripheral proteins, when we talk about peripheral protein, it is partially protein. Its function as an enzymes, and membrane carbohydrates is the membrane carbohydrates it contain is glycoprotein, glycolipids, and proteoglycans. So glycoproteins are membrane containing carbohydrates in combination with proteins. Then glycolipids, membrane containing gly means carbohydrates, lipids means fats. So it consists of carbohydrates with lipids, and proteoglycans is the membrane carbohydrates that bound to a small proteins. So it is proteoglycans. This is the complete outlook, and the function of these membrane proteins is basically the secretion, maintains cellular activity, maintains shape and size, transport, and it is selectively permeable. Selectively permeable means not entire thing goes through it, but only selective, like selective in the sense of only particular particles, like the size of the particle is, mentioned कि only this range of particles can go through. That means the selective permeability. So these are the things which you need to remember. So when we talk about glycocalyx, so in the glycocalyx part, the only thing you need to remember is that it is made up of glycoproteins, glycolipids, and proteoglycans. And its function is cell adhesion, acts as receptors for binding of our hormones, and hormones such as insulin. We have already seen the insulin, like complete pathway of insulin metabolism. So go and watch over there. And cell adhesion, we say, provides adhesion between the cells. So just memorize these things, whatever I have put in the slide. These are the only things which come into your exam. Now moving forward to the cell organelles. When we talk about the cell organelles, and that is to endoplasmic reticulum, we say it is the membranous bound organelle. The second thing we need to remember is that a cisternae. So it has cisternae, whereas in mitochondria it has cristae. So here cisternae, here cristae. Do not forget these two terms. In examination, usually we get confused between these two terms. Then this, what we need to remember about endoplasmic reticulum is that endoplasmic reticulum is of two types, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. RER have ribosomes on the outer surface. So we say it as RER. When there is no ribosome on its outer surface, we call it as SER. Then RER consists of very flattened cisternae. It is very flat. They are continuous with the outer membrane of nuclear envelope. They normally have polysomes on their membrane. So these things you need to remember. Then we come to its function. So the function of primary function is to segregate the proteins, not destined for the cytosol. Its more function, we can say, synthesis of the phospholipids, and assembly of multi-chain proteins or help in the protein secretion. What type of protein secretion? Pancreatic SNR cell. What pancreatic SNR cell secrete? That is digestive enzymes. Fibroblast that secretes collagen, and plasma cell that secretes are immunoglobulins. So just remember these three functions about this. When we talk about smooth ER, so in this we say that in RER our cisternae is flattened, but here we have a tubular and it appears as a channel. Okay. Then its function is to synthesis of the steroids secreting cells. It is the site of detoxification processes. What are the detoxification processes? That is coagulation, oxidation, hydrolysis, conjugation. So these are the detoxification processes. Write it down, it might come. I haven't mentioned over here. Then it play a role in cardiac muscle contraction and contain enzyme for glycogenolysis. When we talk about the mitochondria, the only thing that is like we know since our very childhood, that is, it is the powerhouse of the cell. Mitochondria is lipid bilayer protein structure. Important point: the inner membrane fold of mitochondria folds to forms cristae. Do not forget here cristae, here cisternae. Cristae is formed by inner membrane folds. So mitochondria, we have already discussed entire carbohydrate metabolism. We can now blindly say that mitochondria involves in a TCA cycle, in our beta oxidation, that is our electron transport chain, coenzymes, adenodiphosphate, inorganic phosphate, mitochondria, DNA, RNA, ribosome. So its metric consists all these things, and when we come to its functions, so functions are similar. The functions that mitochondria contain the enzymes that are responsible or that are take place in our respiratory chains, or we say electron transport chain, citric acid cycle or TCA cycle, beta oxidation of fatty acid, and oxidative of phosphorylation. It also produces ATP, that is the energy. That's why we call it powerhouse of the cell. It is essential in many chains, and it contains, since we say that mitochondria consist of DNA, and hence it take part in the synthesis of proteins and RNA synthesis. If you see the lecture of insulin metabolism, you see the importance of mitochondria over there. We see how with the help of this RNA molecule that are insulin and C-peptide is formed, if you remember that part. Moving forward, we can say one more thing in here. Just mention this point. I have forgotten to mention. I have mentioned, sorry, that is mitochondria self-replicating. How it is self-replicating is because mitochondria has its own DNA. So by replication, one mitochondria can form multiple mitochondria in the cell. So mitochondria has a self-replicating property. Moving forward, what is the Golgi body? So Golgi body, basically we know that it is a package. It used to pack the protein released by the ribosomes. It helps in the formation of our glycoproteins. What is glycoproteins? The combination of our carbohydrates with proteins. Then it synthesis acrosomal cap, acrosome. Like if you relate with its sperms, that the sperm has our acrosome, and then acrosome has a Golgi apparatus, and that acrosomal cap then take parts in the reproduction or in the fusion. So it also helps in storage of secretory products, and in Golgi apparatus, we say the lysosomal enzymes are modified and packed as a lysosome. So these are the important points which you need to remember. Do not forget them, because these are the only points which anyone can ask you. Since you are a medical student, so these points are like direct MCQs. So do not forget these things. Now, moving forward, another organelle which we are taking, that is lysosome. When we talk about lysosome, we say it is a digestive system of the cell. Why lysosome? Lysosome is very important because it is considered as the suicidal bag of cell. So important points to remember in lysosome is that it consists of 40 different hydrolytic enzymes. Okay, so basically lysis means solution and soma means body. If we cut this, like we segregate this, so this lysis means solution and soma means body. So it has a 40 different hydrolytic enzyme, but in further we see we also have an oxidative enzyme. So lysosome is concerned with hydrolytic enzyme and not oxidative enzyme. Do not forget this. It's mainly present in phagocytic cells of our body. Another point to remember, then it is of two type. What two type? Primary as well as secondary cells. Now, if we say what are the phagocytic cells? So if you can recall it, we can say it is neutrophils, monocytes, macrophage that usually destroys our microbes. When we concerned about our immune system, we go towards this lysosome, lysosomal enzymes many a times. So mainly present in phagocytic cells. What phagocytic cell? Macrophages, neutrophils, and monocytes. Just remember these three. Primary lysosomes. What are primary? That lysosomes that have not entered into the digestive events, whereas the secondary lysosomes are formed by our phagosomes as well as the primary lysosome. So when we say the equation, we make equation like phagosomes plus primary lysosomes. That's the thing you need to remember. And what is the phagosome? Phagosome is basically the vesicles which is formed around a particle engulfed by phagocyte by the process known as the phagocytosis. So we say phagosomes plus primary lysosomes form our secondary lysosomes, and this secondary lysosome enters into our digestive events. Its function is very easy. That is, first, what they are basically do phagocytosis. What is phagocytosis? They phagocytose, or they engulf the bacteria that are harmful to us. Then it damages cellular structures. That is autophagy. What is autophagy? Suppose a lysosomal function. Lysosome is concerned with the removal of non-functional organelles, or if we have an excess cytoplasmic structures in our cell, then there is elimination process taking place. We called it as autophagy. Auto means self, phagy means eat or removal. So autophagy is the function of lysosome. Now, another thing, lysosome is known as the suicidal bag. We have already mentioned why it is known as, because lysosomes are ruptured and the lysosome enzymes digest the whole cells. Because they digest all the cell, it is known as the suicidal bag. And when it digests the cell, or we say when it digests the whole cell, then there is a deficiency of oxygen takes place, and that leads to suffocation, and it ultimately leads to suicide. So it is known as the suicidal bag of cell. So we have already done with these, and we say autolysis means self-destruction is also done by it. So here lysosome has the hydrolytic enzyme. Again, I'm saying, and the lysosomal enzymes, what are the lysosomal enzymes are? These are synthesized in our rough endoplasmic reticulum and packed by lysosomes in Golgi complex. So we have done complete lysosome part. Another important thing, the lysosomal enzymes which we already know is ribonuclease, phospholipase, glycosidase, collagenase, deoxyribonuclease. Then we have a phosphatase. These all are lysosomal enzymes. So these are very common lysosomal enzymes which we need to know. Another one is ribosomes. So when we talk about important topic, that is our ribosomes. So ribosomes, we already know the units and every basic of it. So we say it is a small electron dense cell. Then it is basically about 30 nanometer in size and are involved in the protein synthesis. Protein synthesis is done by ribosome. 1 mark MCQ. Its structure consists of protein and ribosomes, RNAs. Type, it is ribosomes which get attached to the rough ER, and we have a free ribosome. So RER, the ribosomes which get attached to rough ER, it usually synthesizes the transport protein, secretes proteins, as well as most proteins. Those are stored in the Golgi apparatus, lysosome, as well as endosomes. They're also synthesized by the ribosomes that are attached with RER. Another is free ribosomes. Free ribosomes usually synthesis are protein found in peroxisomes and mitochondria and the cytoplasmic proteins. What are cytoplasmic proteins? It is the hemoglobin. So this is what you need to remember in ribosome. Here you see the picture diagram of the ribosomes. Then we come to another one, that is peroxisome. So peroxisomes contain oxidase enzyme, whereas lysosome contain hydrolase enzyme. So difference between both, now it is very clear to you. So oxidase enzyme helps in the detoxification of toxic substances, contain oxidative enzyme, and it protects cells against hydrogen peroxidase. These 3 points you need to remember when we talk about. So basically the difference can come between lysosome and the peroxisome from this. Another is cytoskeleton. Cytoskeleton is simple, that is complex network of protein. It determines the shape of the cells, and it plays an important role in the movement of organelles and the cytoplasm because it provides locomotory movement. What locomotory organs? Cilia, flagella, filaments are all amongst these only. Now, what are the components of the cytoplasm? It is very easy, that is microtubules, microfilaments. Microfilaments are actin filaments, and one more, that is intermediate filaments. So these are the microtubules. Microtubules, they provide support and shape to the cell. Then it participates in the intercellular transport of other organelles. It assists in cellular movement by forming cilia, flagella, and centrioles. When we see that splitting of spindles at the metaphase, then we come to know what are these centrioles, centrosomes, chromosomes, everything, and it forms the spindles. Once it forms spindles, these spindles usually moves the chromosomes in the mitosis process. What is microfilaments? Microfilaments participates in muscular contraction. Then it participates or helps cells to perform endocytosis, then exocytosis, phagocytosis, complete engulf of the particle. Movement of flagella. It also helps in the movement of cytoplasmic components. And last is intermediate filament, that basically plays a major role in smooth muscle cytoskeletal components. We already know what is actin. Actin is the component of microfilament. I have forget to mention here actin. So just mention actin is it's a microfilament component. It is most abundant in the human cells, and when we talk, it is present in muscles as well as in all other cells. These when we talk about the function of the actin, it is similar to the microfilament. Same. Endocytosis, exocytosis, phagocytosis, the same thing. We are done with this. Then we have the intercellular communication. One thing, we have intercellular connections. So when we talk about intercellular connection, we have our tight junction, we have gap junction, we have desmosomes, zone adherence. So these we see when we talk about the membrane physiology. But here we need to just remember these intercellular communication that is done by gap junction, neurocrine, paracrine, autocrine, and endocrine. So what is gap junction? The junction, it is a type of a junction by which molecules are transferred from one cell to another. I don't have a pic of gap junction. So, okay, you can see over here that it is basically the transfer of one of molecule from one cell to the another. It is seen in our epithelial, cardiac, smooth muscles, and neurons. And the unit of gap junction you need to remember is connexons. And the connexons is made up of connexins. So these few points, just write it down. I haven't mentioned because it is very basic. But we see this when we talk about our membrane physiology in deep, so do not bother about it. So moving forward, when we talk about these, so gap junction, when we talk about the transmission of messages, it is very directly, we see it is directly going on. When we talk about our neurocrine, neurocrine is basically concerned with our hormones part. Okay, so it is done by our synaptic cleft. I haven't put the picture over here, but it is basically through a synaptic cleft. We know pre-synaptic part and post-synaptic part. There we have a gap which is known as the synaptic cleft, where our acetylcholine is released, and then the transmission of messages is going on. Then we have a paracrine and autocrine. So in paracrine, we see there is a diffusion over here. In autocrine, we say it is a diffusion over here. And endocrine, it is circulated in our bloodstream, that the hormones are circulated in the, you see in the bloodstream, these hormones get circulated. It's local or general, it is local. Neurocrine is local, paracrine is local, but diffuse, and endocrine is general. Whereas if we talk about specificity, so it is anatomic location. Neurocrine is basically receptors. We see we have many receptors, neurotransmitters when we talk about neuromuscular junction. So you can watch that video also, it get clarity over there. We have paracrine and autocrine, that is also concerned with our receptor. And then we have a endocrine, that is also concerned with our receptors, that is hormones. So if we talk about broadly what is this neurocrine is all about, so basically in the neurocrine, neurotransmitters releases at our synaptic cleft and act locally to control the cell functioning, we see. And the chemical messenger is our neurotransmitters. In endocrine, our chemical messengers are hormones, and the specialized cells release hormones in the blood, which basically influence the function of the cells at another location. So like hormones work on the targeted tissue. Then we talk about our paracrine. So paracrine chemical messengers can be our histamine, serotonin, vasoactive substances, like these are vasoactive substances. Then we have cytokines. Basically in the paracrine, what happens, cells secrete substances that usually diffuse. Here we give the term diffused in where in the extracellular fluid, and affect neighboring cells. Then autocrine. In the autocrine, we say similar cytokines and vasoactive substances here also. And the similar stories going on. So these are the things which you need to remember in this complete basic general physiology, and the MCQs come from these base things which you might forget at the time of your examination. So just go through this lecture once, open your textbook nearby you and just read it out by watching this lecture, and you see your complete general physiology is completed within 15 minutes. Just go and try. So guys, that is all from my side on this lecture. If you like this series of a one shot, do put your reviews, at least put the like button so I come to know that it is actually benefit. So if you like the content of my video, do like, please subscribe, please share, and press the bell icon so you get notified whenever I put the video. But guys, please like at least the video. It gets me motivated to put more effective content for you. So please do this much at least. And thanks for your support. We'll meet in the another lecture with another topic. And if you like this series, do let me know. I'll continue in this entire 1 week for biochemistry, anatomy, physiology, neuro part, and everything if you find it helpful. So do let me know after watching this video. Till then, take care. Bye.